So if you run SE3 from 8 to 12 clusters, this is what you should see in your web browser. So now we have, instead of having 1K, we have a range of Ks here from 8 to 12, right? Then we have several tabs here, which I have shown you before. Consensus, silhouette, expression, markers. I didn't show differential expression. You can look at it yourself. There is also stability tab, which calculates stability of the cluster. So it will tell you how stable these clusters are when you change K from 8 to 12. So it looks at different Ks, and if your cluster appear, the same cluster appear for different K, it will be stable. If your cluster splits when you change K, then it's not stable. So that's what stability shows you. Also, you have pheno data here, which I should change because it's not called pheno data anymore. So this is a call data of your single cell experiment data set. So these are columns of your annotation, cell annotation table, right? So you can visualize any of this on your heat map by ticking the box. So if we say we want to look at cell type 2, click on it, and it will visualize cell types 2 on your heat map. If you want to look at cell type 1 at the same time, it will visualize cell type 2 and cell type 1. You can also look at the total number of features. So you can, for example, if you think that some of the clusters are biased by the number of expressed genes in the cells, you can also see that, for example, this cluster, you have more expressed genes in these cells than in any other cluster. So it's kind of useful to have everything from quality control, which was done by Skater, on your heat map as well. So the silhouette plot is what I showed you before. So this is the stability tab. So stability is calculated. So it tells you that cluster 4, 6, and 9 are not stable. So if you change K, these clusters will most probably disappear. They would split into more clusters, or they will merge with something else. The other clusters are quite stable. Differential expression is similar. Well, it's not marker genes. For the marker genes, SE3 defines marker genes as genes that are specific to a single cluster. So these genes are more expressed in one cluster than in the other clusters. Differential expression is more general. It looks at differences between any two clusters. So if you have one cluster which is different from anything else, then these genes will be called differentially expressed. So here you will see this gene is expressed in several clusters, but not expressed in this one. So this gene will be called differentially expressed. Obviously, differentially expressed genes that are differentially expressed contain all the genes that are marker genes. Because you can, this way you can also define marker genes, but marker genes are more specific to the cell types. And we can, as I said, we can change K. So 8, 9, and you can see how your structure changes. So this 10 is actually good because here you had lots of yellow color off the diagonal, which you don't want to have. So what you want to see is only red color on the diagonal and only blue color off the diagonal. So if you go to 11, 12, probably 12 is even better than 10. But we didn't run it for 6, so if you run it for 6, probably the consensus structure will be the best, I suppose. No? All right. So, but again, this is once you calculated clusterings for different Ks, then it's your subjective decision which one to choose. So it's not, there is no unsupervised way to define which K is the best. Okay, so let's continue. And move on to the next tool, which is called PCAreduce. Yeah. Okay, so I didn't show that. So to run SE3 for multiple Ks, you need to use in the function call, instead of single K, you need to use a range of Ks from 8 to 12, right? So it's 8, column 12. Okay, so let's move on to PCAreduce. So PCAreduce doesn't integrate single cell experiment objects, so we need to export the expression matrix first and then run PCAreduce on the expression matrix. So here we say we define this variable called input. We take log counts of our data set, and also we remove some of the genes. So I didn't cover that in SE3 chapter, but SE3 also has some gene filter, which is very general. So it removes all the very, very lowly expressed genes in your data set, and also it removes the genes that are expressed everywhere. So these genes do not contribute to the clustering solution. So we basically do some gene filtering and export log counts of our data set. So I think here we should have a list, but somehow there is not list. So PCAreduce has several parameters: NBT, Q, method, and S, and M. So there are quite a lot of parameters. You can look at the documentation. Here we use, I think, some default parameters that are described in the paper. Also, we need to transpose the expression matrix. So that's another problem. And some clustering tools use transposed expression matrix, so they assume that you have cells in rows and genes in columns. Whereas in single cell experiment, we have cells in columns and genes in rows. So when you run different tools, you sometimes need to transpose your matrix. So in SE3, because it's iterative, I think Q defines the initial number of clusters, and then it finds hierarchy of clusters from Q to 1. So it will find clustering solutions for Q equals 30, 29, 28, and so on. But so there is I had to spend some time here. So probably this is not the number of clusters, but basically Q equals 30 corresponds to K equals 1. Q equals 29 corresponds to K equals 2, and so on. So if you want to have 10 clusters, we need to subtract 10 from 32. I don't remember why 32, but you can believe me. You can look at this variable yourself and see how it looks like. But yeah, it was a bit complicated. So yeah, we write this clustering solution to our single cell experiment object, and we call it PCAreduce. So then we can plot PCA again and color cells by this clustering solution. That's what we do. So as you can see, it's actually not too bad. So we have kind of reproduced the stages of embryo development here. This is a bit mixed, and this is a bit mixed. So it looks okay to me. So we have an exercise here to run PCAreduce for k equals 2 and see how it looks like. So that's how it looks like, which also makes sense. So when you look for two groups in this data set, probably you can merge this ones and leave this one as a separate group. Another exercise is to calculate adjusted rand index, the ones that we used before. If you do it, you will find out that your adjusted rand index is actually lower than for SC3. So the solution is a bit less accurate if we compare it to original cell types defined in the publication. Any questions here? So sometimes if you want to be sure that you get reasonable clusters, you might want to run different clustering tools on the same data set and compare the solutions. If you get consistent clusters, then you can be sure that your solutions are okay. The next one is t-SNE plus k-means. This is just, we're doing it just for fun. So if we plot t-SNE using plot t-SNE function of scatter, this is how it will look like. So all points on the plot are black because we don't use any cell type annotation. We didn't ask to color the cells. But because we run t-SNE and we also use the parameter here called return SC equals true, when we use this parameter, scatter will write the results of t-SNE dimensionality reduction into reduced dimension slot of your single cell experiment object. So then you can, or maybe, is it? Yeah, I think so. So here, we write the results of t-SNE again to our cell annotation data frame, right? And we call it t-SNE k-means. And the way we write it is this. So as I said, the results were written to reduce DIMS slot of our object, and it's called t-SNE. So this was created when we run the previous function. So when we run this with this parameter, it creates additional slot and writes the results of t-SNE. So then you can reproduce this plot when you need it. So we actually start with k equals 8 here. Okay, so we also run k-means, right? So this is t-SNE plus k-means. So we take the results of t-SNE, and then we run k-means on it with 8k, k equals 8, right? And then we get clustering solution from that. Because t-SNE itself doesn't do clustering. It just embeds your data in 2 dimensions. So you have coordinates of your cells, and then you cluster them by using k-means. So if we ask for 8 clusters, this is what we get. We also need to see what are the cell types here, right? So if you run the same function but color by cell type 2, you will see whether the clusters that were found using t-SNE are the same as the cell type 2. But if you also run adjusted random index, it will be 0.39, so which is even lower than PCAreduce. So the clustering was not really good. And this is what we expect with t-SNE. It shouldn't be, it's not the best method to use for clustering. Any questions? Okay. So the next method is called SNN-click. This is a graph-based method. You also need to define several parameters. So we provide, so we have this package, SNA, single cell RNA-seq functions. This is our package, and we provide a function SNN in this package, which makes it easier to run SNN-click. But you can go to the website of SNN-click and run it manually. This tool is actually, it's a Python-based tool, so we need to run Python to be able to get the results. So we run Python from R session. You can do it in your terminal if you wish. Yeah, so this is a bit more complicated. So some tools you can only run in Python. So you will need this file to run it. So this is the file which the developers provide for SNN-click. So what it does, this is the output of the tool, and what it says that it finds 29 clusters. So SNN-click is the algorithm. Actually, it finds K itself. So it only provides a single solution, and it will always find some K. So you either trust it or not, right? So this tool found 29 clusters. Now, if we read these results, save them into our single cell experiment object, and then we can visualize it. For this one, I think you don't need to set the seed. This is deterministic, so you shouldn't, yeah. So now we can visualize the results of SNN-click with 29 clusters. It's quite messy. So this tool probably fails to find good clusters in this data set. And adjusted random index is 0.26, so it's the lowest one. The next tool is sincera. As I mentioned before, it's basically just simple hierarchical clustering. But in addition to that, it has some functionality to calculate K. So the K is calculated using the following idea. So K is the minimum height of the hierarchical tree that generates no more than a specified number of singleton clusters. These are the clusters containing only one cell. So it looks at it creates the hierarchy from your cells, and then it goes down from in the hierarchy and finds a level where you get a cluster with one cell, and that's how they define K. They stop at this point and they say this is our K. So once you start getting one cell in one cluster, this is the stopping point for sincera. So actually, if you do that, sincera also finds 6 clusters, like SC3. But this is not, we don't recommend using this approach. So sometimes you might get one cell in one cluster because you have a rare cell type, but then you will miss everything else afterwards if you don't go down, increase the K. So we can visualize the results of sincera. So this heat map looks a bit different because the genes, when you run sincera, you normalize the genes in the beginning. So the values of the genes are, I think they're from 0. Oh no, actually here you have from 0 to 6. So, but there is some normalization, so they are, the values are more comparable to each other. And if we run adjusted random index on sincera, it's also quite slow. So it's 0.38, even though it found 6 clusters. So it was you would think that if it found the correct cell types, the adjusted random index should be higher, but probably the clusters themselves are not ideal. You can investigate and plot, visualize the cell types on top of the heat map if you want to, as an exercise. Any questions? So obviously nowadays there are a lot of clustering algorithms for single-cell RNA-seq data. Here we cover only a few of them, which we think are important, but every month you have another algorithm published. So keep an eye on what's going on with clustering algorithms for single-cell data. Yeah, and I hope you got some idea of what you can do.